Hello, everyone. A warm welcome for all of you for today's webinar session we have. And today we are very much overwhelmed and we are honorable to invite our speaker, Dr. Sheela Sasikumar. And on behalf of Organogenic, we are going to present a webinar on topic translational research from bench to bedside. Before going to our webinar session, I'm going to present a small introduction about our honorable speaker. We have been overwhelmed with our CV, but even though unfortunately, as we have very less time, I would like to mention very few of her achievements. She is a PhD FSc SC Director Clinical Research at Hycare Superspeciality Healthcare Hospital. She is an eminent clinical research specialist having rich experiences spanning over 3 decades in the field of clinical biochemistry, molecular biology, and natural products bioactivity. She has been member in various leading institutions and won numerous awards in biosciences, which we can't even believe. So just to mention one of our prestigious awards, I would like to mention award Professor E.P. Odum Gold Medal 2007 for commendable research activity from Scientific Advisor to Prime Minister. She has over 50 publications in reputed national and international journals. We are very warm welcome, ma'am, Dr. Sheela Sasikumar, to take up the stage to present the webinar. Thank you.
Thank you, Vaishnavi, for your warm introduction. Hi, everyone. Good evening to all. Welcome to the webinar session, A View on Translational Research from Bench to Bedside. At the onset, I wish to thank Golden Jubilee Biotechnology Park for Women and Organogenic Biotech for providing me an opportunity to share my views on the translational research from bench to bedside. I also wish to thank Dr. Sudhakaran Iyer, the GM, Biotech Park, and also the organizers, the Gaurav and Shamini. Thank you all. Translational research from bench to bedside. So I'm working in the clinical research department as a consultant in Hycare Superspeciality Hospital and also in SS Healthcare. Please watch my channel, SS Sheela, for the recent webinars on COVID-19, immunoboosters, and immune activity. So this is my consultancy, and we are into the research trials, publication, workshops, patents, and all in one umbrella. Please use these links to view my research article. I have nearly 56 research publications. So all my research articles are shared in these platforms. So translation research, what are the topics I'm going to cover in this part is introduction to translational research, how translational research is acting as a bridge between the basic and the clinical research, translation spectrum, key step for success, preclinical and clinical trials, regulatory bodies, and IPR, protecting the IP rights. So to go about, I have been impressed with the urgency of doing. Knowing is not enough, we must apply. Being willing is not enough, we must do. Leonardo da Vinci. This impressed me a lot in translational research. So what is translational research? A translational research is a long journey starting from bench to bedside. The journey moves from the scientific lab ultimately to the hospital, giving benefits to the community and to our people who suffer from specific diseases. It can be either a drug or medical devices or a diagnosis, anything. So this picture I have shown here shows you many problems surrounding us. So we have many problems, existing problems. Right now we have the COVID-19 pandemic situations. So many pharma, many scientific organizations, scientific labs are proceeding with different vaccines, kits, drugs, etc. to find a solution for this pandemic problem. So now we are going to relate this. What is research? So you have number of problems in the environment. So the scientist has to select a problem. So the first part of your research is going to be a selection of a problem and finding a methodology of a research. So if I have selected a problem, I should have a standardized methodology and it should be clearly spelled out. Once I find a suitable methodology and execute the research, the next step will be the collection of data and analysis of my data that is obtained in my research. Then interpretation of my data to produce my inference of my research. So this entire pathway is called as a research. Research normally is of two types, basic research and applied research. Basic research, it is nothing but a desire to expand the knowledge, curiosity driven, intent to answer why, what, or how the questions, increasing understanding of basic fundamental principles, but does not have an immediate commercial objectives. It may not necessarily result in an invention or a solution to a practical problem, but it answers only the fundamental questions. But the basic research leads to new products, technologies, and processes. Applied research. So as the name suggests, it's applied. A new knowledge which is acquired has specific commercial part. So it has a commercial value. The objectives can be products, procedures. You have invented a new protocol or diagnostic kit or any type of services. So it answers, your applied research gives answers to the specific questions aimed at solving the practical problems. So here we are going to create new products, technologies, and processes. So leads to new fundamental questions. So these are the two types of research that happens in the community. Basic research is performed without thought of practical ends. It results in general knowledge and an understanding of nature and its law. This general knowledge provides the means of answering a large number of important practical problems, though it may not give complete specific answer to any one of them. So basic research, as I said, you can create a knowledge, you can do many practical solutions for it, but not necessarily it can give you the complete answer to any one of them. But whereas the function of the applied research is to provide such complete answers. So translation research happens to be a bridge between the basic and the clinical research. So what is translational research? So translation, it is translating the basic research into a form of research that becomes beneficial to the community. So bench-to-bedside process that harness knowledge from basic scientific research into clinical research to create novel treatments, any new treatment option devices, medical procedures, preventions, diagnostics, essentially forming a bridge between basic research and a clinical research. So it is going to create a bridge and entirely translates into a new type of, for example, a device that will help in the medical sector or a health sector, either to find out the virus, for example, or detect the virus in the blood, in the saliva, anything. So translation research, a new area of investigation that ideally involves the integrated application of innovative technologies. So I here represent as innovative technologies, new technology that encompass the multiple disciplines including physiology, pathophysiology, natural history of disease, genetics, proof of concept of drugs and devices. So normally this translation research includes two areas of translation. So it is going to translate in two ways. What are they? One, the process of applying the discoveries generated during the research in the laboratory and in preclinical trials to the development of trials and studies in humans, that is preclinical trials to clinical trials. The second area of translation concerns or the research will be aimed particularly at enhancing the adoption of the best practices in the community. Cost effectiveness of prevention and treatment strategies is an important part of translational science. So during this journey, whatever the basic research, we convert it into a preclinical trial to clinical trial. Finally, the best will be adopted, which becomes beneficial to the community. So now we know what is translational research, how long it has to travel. So when a new drug enters into the market, it takes more than 6 years to 8 years or even 12 years. So what is the path that it is going to travel across? So this entire pathway is called as the translational spectrum. So this translational spectrum involves many phases: T0, T1, T2, T3, T4. T0 is introduced recently. So these phases, if you see the picture, you can see the different phases. The definition for the translation spectrum given by FDA is translation from the basic science to human studies form the critical path or valley of death. Valley of death is defined by pharmaceutical industries. This valley of death encompasses the T0, T1 and T2 phases. So to cross these two phases, you have to face many hurdles. So this valley of death is very important in the translation spectrum. So we can see individually each phases. Now regarding the T0 phase. T0 phase is now introduced by US Food and Drug Administration. It is nothing but a micro-dosing study involving 10 to 15 number of subjects, that's all. So let us see the T1 phase. So translation to humans. So translation to humans. So when are we going to translate? After getting the preclinical studies, results from the animal studies, then it will be introduced first in the human studies. So T1 will become first in the human studies, that is called phase 1 human trials, which will be see later in clinical trial. T2 is translation to the patients. So phase 2 and phase 3 clinical trials will be involved. T3, translation to practice. Once you translate to the patients, when you get the results, pleasing results, satisfying results, and when you succeeded, it will be translated to practice. So translation to practice involves phase 4 clinical trials, dissemination and implementation of the research. T4 phase is nothing but translation to population health, that is studying in a community, a large number of people. The population outcome studies, this phase will show the true benefit to the society of this translation research. So as I said, valley of death. The valley of death as defined by the pharmaceutical industry encompasses T0. If you see here, it is this valley, and this valley is very important. So it encompasses the T0 and T2 phases of the research. So this translation continuum should proceed. So this valley of death should be crossed by a group of people who are going to convert this basic research into a translational research. So what should be done? The main thing is we need to remove the physical barriers that is blocking our collaboration. So basic research people or basic scientists have to collaborate with the translation scientists, and they should work on together. Provide access to adequate funding, infrastructure, everything is needed. So the access to these should be provided. Only then the valley of death will be prevented and they can cross the hurdles. So if you see this picture, this is the entire map of the translation research. I have given you the hyperlink at the base of the slide. You can see this hyperlink. It's an NCBI thing where this translation research map is provided. So this explains from the fundamental research, that is starting from the basic research to the different phases of the translational research, and how the data get transferred to produce a device or a drug or any type of service that becomes beneficial to the entire community. Now, if you see the data transfer here, from beside the bench, what happens? Creating knowledge. So knowledge is created. The basic scientists, they find out a problem, they start working on it, they find a suitable methodology, they execute the research, the analysis of the research, the data collected will be interpreted. So the creating of knowledge, testing the knowledge, interpreting the knowledge. Then finally what happens? Whatever the knowledge they have found out will be implemented if it is following standard procedures and protocols. And this knowledge impact. So this becomes a data transfer. So during this travel, what are the gaps that can be found? So normally if you see between T0 and T1 phase, there is a gap between the fundamental research and developing a medical product. Sometimes all the basic research cannot be taken into a clinical trials. Sometimes the problem may not be the same that exists in the medical community. So right now the problem is going to be COVID-19. So we should work on a research that is essentially needed for the beneficial to the people in preventing the COVID-19 or coronavirus. So there can be a gap between the fundamental research and developing a medical product. Between T1 and T2, there can be a gap between developing a medical product and making it clinical. Okay. So between T2 and T3, there is a gap between making a medical product clinically ready and its routine application in clinical practice. Sometimes what happens, that clinically it is ready, but to practice it, it may take time. So there becomes a gap. There is a time lag in implementing it as a clinical practice. So the gap between T3 and T4 is the gap between the routine application in clinical practice and its population-based effects. So once it comes into clinical practice, how many people or the community they get benefited. So you have to get the data. If it is not available to the entire community, then there is no use of this translation research. So between T4 and T5, there is a gap between the population-based effect and policies derived from it. It may take time because the regulation, you have to come across many regulatory bodies, etc. So this map entirely represents the hurdles, the gaps, and the data transfers across the phases. So finally, what we should understand is there is no I. The pronoun I has to be removed. It is only we. If you want to go fast, go alone. If you want to go far, go together. So this is very, very important in case of translational research. A multidisciplinary approach and a teamwork is highly essential to strengthen the research and to develop it into a beneficial product. So what are the characteristics of a translational scientist? So what is needed? One, boundary crosser. So you should break down the disciplinary silos and collaborate with others. So I said you should not take the pronoun as I. Whatever I am doing is correct. No, you have to discuss with the multidisciplinary team. So your team will have a physics, a chemistry, a biotech person, a genetic person, a physiology. So a multidisciplinary team should be created. So you have to create that and profession to collectively advance the development of the medical intervention. So whatever the ideas have to be discussed and then it has to be proceeded. Then you should be a good team player. Team player is very, very important. So valuing the contribution of all the players. Each one will contribute. In a multidisciplinary team, each one will contribute a part of the invention. So you should play well, respect everybody's creation and bring it together as a combined form. Rigorous researcher conducts research of highest levels, transparencies, possess strong statistical analysis. Statistical analysis is very, very important in order to create it into a better product. So reproducibility has to be done. Maximize the reproducibility. Process innovator seeks to better understand the scientific and operation principles. Domain expert. So you cannot be a domain expert in all areas. In your area, you can be a domain expert and you can discuss your innovations with the other disciplinary team and get a better understanding to go through the journey. A very good communicator, a skilled communicator, communicates with understanding with all stakeholders in the translational process across diverse social, cultural, economic, and scientific backgrounds. So, whenever time permits, you have to share your innovation with the different bodies and with the investors, with the sponsors to take the translation process across. Then you should be a good system thinker. So these are the characteristics of a translation scientist. So translation, we said it is a process you are going across the scientific lab into preclinical trials, clinical trials, and ultimately into the community. So during this process, two things has to be remembered. One is GLP, good laboratory practice. A set of principles. What is GLP? It is a set of principles that provide framework within which the lab studies are planned. So you should have a GLP lab that is going to plan, perform, monitor, record, and report, and all your details should be archived. Another thing it have to be taken into mind is GMP, good manufacture practice. It is a system. Products are produced according to the quality standard to minimize the risk involved in any pharmaceutical production. So GMP facility is very important to create a quality. So now we'll see what are the key steps for the success in the translation research. The first key success is identify a unique area based on your available expertise and your interest. So identification of unique area is the first key point of success. Number two is you have to establish a collaboration with the experienced researchers in the field. So you should share the knowledge and your invention with them. Identify a unique area on the available expertise. I think it's repeated. Laboratory experience is essential. So you cannot say that you do this, you do that. You should have the experience. For example, if you want to do some molecular biology techniques, you should know all the process, including RT-PCR and all the developing process has to be known. So build a team. So I said you should build a multidisciplinary team, which is very, very important. Present your work often to the collaborators, either in the individual meetings or small group. This is very, very essential. Become a part of the research working groups at your institution. Always take small steps. Don't take large steps which will make you to fall. Take small steps. Be creative and fertile. If you follow these steps, I think translation research can succeed. Developing a novel drug in an interdisciplinary endeavor involving multitude of competence from biologists. You need a biologist, you need a chemist, you need a computer scientist, that is bioinformatics people, medical staff, biostatisticians, and also regulatory experts. So these are the some areas where you can create a multidisciplinary team for a development of a new drug or a novel drug. Taking a compound from a bench to bedside requires up to 12 years at an average estimated cost exceeding US 1 billion. So if you see the picture here, you can see the basic research, target identification, target validation, drug discovery, lead identification, lead optimization. So preclinical research. So out of 5,000 to 20,000 compounds come in this particular phase. When it comes to preclinical research, only 250 compounds are coming. So they undergo pharmacodynamics and pharmacokinetics, toxicology studies, safety and efficacy studies, etc. Out of 250 compounds, only 5 compounds will enter in the clinical trial, phase 1, phase 2, phase 3 studies. Out of that 5 compounds, only 1 compound will be approved by the regulatory body and it will be marketed. And phase 4, the post-marketing thing will be done. So this is how it comes from the basic research to a approved drug. So the next phase of in the translation research, we completed basic research. The next phase of the translation research is the preclinical trial. So what is a preclinical trial? Why is it essential to do a preclinical trial or execute a preclinical trial? Safety and efficacy of a drug has to be tested before it is introduced in humans. So preclinical trial, the aim or the objective of the preclinical trial is to elucidate the efficacy and the safety aspect of a drug. So in case of preclinical trial, what should be kept in mind is lab test of a new drug or medical devices is usually done in animals. So in case of preclinical trials, we are using only animals. But what should be noted here is only use the drug or devices which is done with the help of GMP. So as I told you earlier, there are 2 things which has to be kept in mind. One is GMP and GLP. So must use GMP drugs or devices because we need quality standard throughout the protocol. The main goal, as I said, is to test the efficacy and the safety aspect of the drug. So usually the experiments are performed on rodents, that is small animals, that includes rat, guinea pig, hamster, rabbit, etc. When the results are proved to be successful in small animals, the same study will be extended to the large animals. So the large animals are going to be cat, dog, monkey, etc. So in preclinical trial, animals are used as a model. So what are the key steps in a preclinical trial? First, you need to identify the drug target. So once you identify the drug target, you must do the bioassays. What are the assays that has to be done? Then screen the drug. Effective and toxic doses has to be identified. Once you come across all these things, once you finish all these steps successively, then what happens? You have to create a file for the approval as IND. What is IND? Investigational New Drug. So this IND will be filed for approval to the regulatory body. So in case of preclinical trials, we take it in two forms, either as in vivo and in vitro. So in vitro, you can do all the tests that is needed, that is given as a standard protocol. In case of in vivo, we come across two phases. One is pharmacokinetics. What is pharmacokinetics? A relationship between the concentration of the drug in the body and its biological effect. So you are going to see what happens to the 5 milligram of the drug which is given to the animal, how it is metabolized, how much is available. So you're going to see the bioavailability and all those things. So that becomes a dose response. So you will study at different doses. The other part is pharmacodynamics. So pharmacodynamics is nothing but it completely gives us the description of changes in the plasma concentration over a time as a consequences of ADME, absorption, distribution, metabolism, and excretion. So these two studies will be done under in vivo. In case of in vitro, as I said, we will do certain tests. So what are the tests we are going to do in vitro with the GLP facility? One, screening test. Next is test on isolated organs can be done. For example, if you want to study about a diabetic drug, so you can use pancreas or the hepatic liver, etc. So isolated organs can be used, or you can use cultures, bacterial cultures, or you can use cell culture. Test on animal models of human disease, observation test, confirmatory test, mechanism of action of the drug, what happens when it is taken orally or when it is given topically, etc. Systemic pharmacology, quantitative test, pharmacokinetics, and toxicity test will be done. So if you see the picture, the drug discovery, this is the target. So from the drug discovery to preclinical development, and you're going to file it as a IND. So investigational new drug. So in case of drug discovery, basic research, custom synthesis, efficacy, in vivo efficacy. Whereas in preclinical development, you're going to see the preparation, formulation studies, safety studies, pharmacokinetics, and ADME studies. And along with, then you're going to file it as a new drug. So in case of preclinical development stages, these preclinical activities provide the basis for the IND application to the FDA for permission to initiate the clinical testing in humans. So this is the hyperlink I will provide it. So once preclinical, the next aspect, once the preclinical trial is successfully completed and IND is sent for filing for approval, what happens is next is to initiate the clinical trial. So after researchers test the new treatment or procedures in the lab and in the animals, which includes small animals and the large animals, the most promising treatments, as I told you, not all the things will be brought into the clinical trial. The most promising treatments are then moved into the next phase of the translation research called the clinical trial. As new treatment moves through a series of steps called phases, more information is gained about the treatment, its risk, and its efficacy. So the next phase of the translation research is clinical trial. A systematic investigation in human subjects to evaluate the safety and efficacy of a new drug. Translate the research or translate the results obtained from the basic research into better ways to prevent or use it as a diagnosis or use it as a treatment for a particular disease. So what is a clinical trial? It is nothing but again a clinical research, a study that is conducted using patient volunteers to find the safe and effective treatment for a variety of health conditions that are critical to the advancement of medicine and improving the patient health. So here you can see how the clinical trials play an important role. So when a new drug comes, so it is tested in the volunteer patients. So you need a efficient and skilled healthcare team. So these clinical trials help to improve the healthcare, and they play an important role in improving the healthcare by helping us to develop new drugs, new clinical procedures, new diagnostics tools. Also, for many new questions for research, which ultimately leads to new discoveries and better understanding of a disease, of a particular disease. What are the different types of clinical trials? Now we know what is a clinical trial. We are going to study in the humans. So what are the different types of clinical trials that can be executed? It can be simple randomization, where the subjects, we call subjects, persons or the people or the individuals who are participating in the clinical trials, we call them as subjects. So here the subjects have equal chance of assigning into one of the two or more groups. For example, in my clinical research, I have two groups. One is a test, another one is control. So I will use simple randomization. For example, tossing a coin. So tossing a coin, I randomize the subjects into two groups. Next type of study is blind or open label. What do you mean by blind or open label? In case of open label, the principal investigator and the patient know about the drug. Whereas in case of blind, where you have two types, single blind and double blind. In single blind, the patient will not know the type of the treatment that is being given to him. In case of double blind, both doctor and the patient will not know about the details of the drug. Step is prospective or retrospective. In prospective study, what happens is that subjects are enrolled before the study begins, whereas in retrospective, what happens is you can take the past case records. So, for example, how many of them have come across the diabetic foot ulcer? What type of treatment is given? What is the statistical analysis of that? So you can get the data and you can use the analysis. So this type of study is called the retrospective study, where you are using only the medical records. Next type is called placebo. Placebo is nothing but dummy drug. It resembles the active drug in physical properties, but do not have pharmacological activity. The next type is pilot study. Pilot study is a very study that becomes the base for a bigger study, where the number of subjects taken will be less than 30, etc. So the study will be conducted. Once the results are pleasing in the pilot study, it will be extended to many people, for example, 100 numbers, 200 numbers, etc. Next is crossover study. In crossover study, two types of treatment are studied in the same subject or same group. So these are the different types of clinical trials that can be executed from the basic research while translating into a clinical trial. So now let us see what are the phases of clinical trial. So as I said, you have preclinical trial, that is you are doing it in the laboratory with the animals. So in case of that, you get the IND after doing so many tests, and you would have come across a suitable dosing study, and you will have a knowledge about the safety, you will have a knowledge about the toxicity, everything comes here. Then what happens is that it moves into the phase 1 of the clinical trial. In phase 1 of the clinical trial, you are going to evaluate the safety, gather the information about how a drug interacts with the human body. Then phase 2, safety and dosing. Phase 3, safety and efficacy. And phase 4 is post-marketing, and how it has been taken into a community. So once phase 3 is over, you go for the FDA approval. So phase 1 trials. Phase 1 trials are conducted in a small group of healthy people, usually 20 to 50 numbers, that the first time to evaluate its safety, to determine its dosage. Phase 2 trials are nothing but extended to patients. So a group of 100 to 300 numbers in order to evaluate its efficacy and safety. So here you are going to see the effectiveness of that particular drug in a disease. Phase 3 trials. Large group of patients are involved, 1,000 to 3,000 number, where we are going to study the effectiveness, monitor any side effects. So adverse side effects will be monitored, compare it to the commonly used standard treatment protocol or standard drug, and it is used, how it is used safely. In case of phase 4 trials, after phase 1, phase 2, phase 3, you go for the approval. And once the approval is done, you go for the phase 4 trial. That is nothing but post-marketing studies, drug risk, benefits, and the optimal usage. So these are the different phases of a clinical trial. Now, what are the regulatory bodies that we should come across when we are translating all these things? First one is FDA, that is nothing but Food and Drug Administration of US. In India, we have DCGI, Drugs Controller General of India, CDSCO, Central Drug Standard Control Organization, which comes under DCGI, and we should follow the guidelines of ICH GCP. ICH is nothing but International Conference on Harmonization. CDSCO is controlled by DCGI, which lay down regulations, rules and regulations to conduct the clinical trials in India. So all clinical trials are conducted in India should be as per ICH GCP guidelines and follow recently amended Schedule Y of Drug and Cosmetic Act. So this is very, very essential if your product has to come to the market. So the clinical trial protocol comes across Drug and Cosmetic Act Schedule Y, which is a complete requirement and guidelines for permission to import or manufacture of new drugs for sale or to undertake clinical trials. It exclusively describes the details of application process for conducting clinical trials. So it also describes the responsibility of the sponsor, responsibility of the investigator, and the role of the Independent Ethics Committee for providing the approval for such studies. ICH Guidelines E6 is nothing but GCP Integrated Addendum to ICH E6 Clinical Trial Protocol and Protocol Amendment. Next is ICMR National Ethical Guidelines for Biomedical and Health Research involving human participants. Here, completely ethical consideration. So all studies have to be approved by the Institutional Review Board as well as the Ethical Committee. Only then it will be extended into, only then the study can be extended into the patients. So the clinical trial protocol, the clinical trial protocol will have the following components. It should have an introduction, there should be an abstract, the objectives has to be clearly spelled out according to the study, the background, the rationale for conducting such study, eligibility criteria, which includes inclusion and exclusion criteria, what are the subjects that can be included for that particular study, what are the subjects that needs to be excluded. Then you have study design methods. So you will have standard methodology for drug as well as for device or for diagnosis, criteria for evaluation, study treatment, clinical assessment, clinical laboratory measurement. So all the laboratories should be certified under NABH. So there is certain assessment for all these things. Then evaluation by visit, how many visits. So from day 0, how many visits has to be performed. Adverse effect. So you have to report the adverse effect and serious adverse effect. Then documentation, which is very, very essential. discontinuation and replacement of subjects, protocol violations, if any, has to be noted. Statistical section is absolutely essential, and this biostatisticians have to do the analysis of complete study and monitor it. Administrative is nothing but ethical guidelines are followed. Do they have the regulatory consideration? All approvals are available. Then you have the subject confidentiality, which has to be maintained according to the ethics. Then references, publication. So this is a simple, complete overview of a clinical trial protocol. It depends on the study. So a clinical trial in a nutshell is represented as a picture. Once I get approval from the protocol or from all the regulatory bodies, from the institutional review board, from the ethical board, etc., then what happens is selection of investigator. The principal investigator will be selected according to the nature of the protocol. So if it is a diabetes or if it is a diabetic foot ulcer, who you are going to select? So the expertise person will be selected as the principal investigator. Then you go for the approval process in that particular hospital. Then what happens once you get the approval, the recruitment of the patient will be done. Recruitment of patient, once done, the study will be commenced. Then the data, every visit of the patient will be entered in a diary and all the case histories will be recorded. Then finally, the data which you have entered will be analyzed according to the statistical rating. Then it will be presented and publication of the report will be done. Once the publication of the report is performed, the data will be filed and the registration will be obtained. So this is a complete picture of a clinical trial procedure or a process. So success of a clinical trial again depends on a team. So where there is no individual person involved here, you require a team. So the sponsor, the principal investigator, the collaborators, the pharmacist, nursing people, regulatory board, data management, that is including the statisticians, will give a successful or will finally evolve into a successful clinical trial. So again, it is a multidisciplinary approach. So here I have shown you a picture in case of drug research and development process. Once you identify an active compound, either from a herbal or synthetic or whatever it is, so the basic research takes as much as 2 to 3 years to show it is an active compound. Then this active compound goes for the non-clinical studies, which again takes 3 to 5 years. Then to clinical studies, 3 to 7 years. Then new drug application and review, approximately 1 year. Then approval and launch. So as I told you earlier, it may take about 12 years to develop a product or a new drug. So here I have shown you a clipping. You can go to the NIH clinical trial site. So where you can see, I have taken specifically, shown here specifically for COVID-19. You can come across the clinical trial which is done for various diseases. All the clinical trials have been registered at this particular site. So you can come across here, this is the particular site from CDC and from NIH. When you go, you can see now you can see the COVID clinical trial done for vitamin D, vitamin C, hydroxychloroquine, and hyperbaric oxygen therapy. There are so many clinical trials performed in different parts of the globe, which will be registered here. So we'll have an idea about a clinical trial. So what are the common hurdles that we face? Normally, what we face is sometimes what happens, the basic research which you have struggled and performed for 2 to 3 years or 3 to 4 years may not be clinically relevant. So it cannot be taken into a translation or it cannot be translated into a active product into a society or into a medical treatment into a society. Sometimes there will be shortage of translational investigators to investigate. Then inadequate collaborative approach. As I said, we need a collaboration with different area of science. So multidisciplinary team should be there. Poorly integrated activity from academia to private sector and industry. Normally we face this. Inadequate financial support, which is absolutely the scientists face this problem. Regulatory impediments to translation, inflexible clinical trial designs. So I have just picked out the common hurdles based on my experience. There can be more than this also. So how to overcome this? Few points that can be taken into consideration to overcome this. Basic science is a foundation of medical advancement. So, investigators with deep understanding of fundamental biology and mechanism of disease are essential to translate the discoveries into new and improved health intervention, diagnosis, and treatment. Additional training, resources, and support would enable the basic scientists to move the discovery forward effectively and efficiently. Research community should expand, this is important, should expand the translational research training communities for basic researchers and training, so that basic scientists will also learn, and they also have an idea of what the medical field requires to facilitate their access to funding, equipment, infrastructure, and other sources needed for translation. So, financial support is absolutely essential. Encourage and support collaboration between the basic and clinical investigators across the research disciplines and sectors. So, this will overcome the hurdles, and ultimately it can be efficiently translated into a new product. So, creating a strong translational research programs are key to combating this coronavirus disease 2019 pandemic or COVID-19 pandemic. By facilitating research that spans from bench to bedside, translational research uses cutting edge laboratory techniques to study the patient sample, immediately pivots laboratory discoveries into new therapies for the patients across the medical spectrum. See, if you see the global statistics, this translation research process from cancer to HIV. Translation research has been critical to reaching our ultimate goal to design new diagnostic tests, the mechanism of the drug or diseases, establishing and improving the advanced therapies. So you should create a strong translational research programs. So today, if you open the NIH site, you can see hundreds of vaccines is produced and tested, some cross phase 2 trials, some cross phase 3 trials. So there are many translation research going on for the COVID-19. So the success of translation from the basic research to bedside depends on adoption of multidisciplinary team science, inter-organization collaborative model that synchronizes efforts and action of all funding agencies, academia, non-profit foundations, philanthropy, pharmaceutical and biotechnology industries, the public and the policy makers, with full support and backing of organization leadership and funding agencies. So that will lead to success from the basic research ultimately into the human clinical research, which becomes beneficial to the patients. So the last part of our translation research is importance of protecting the intellectual property. This is very important. So why to protect our IP? So universities and academics commonly make use of grants. So we get different types of grants from the government to pursue our research, from secondary or tertiary sources to fund the research project. Such funding, however, is generally insufficient to develop a clinical product. So what is the solution for that? Combine an academic research with the elements of corporate business model for pharmaceutical product development. This means IP rights should be established. Once you are collaborating, your IP rights should be established on inventions that have been conceptualized during the course of the research work. So why do you want to protect your IP? The development and implementation of invention is arduous and very costly. So it is crucial to protect your rights. Otherwise, you're struggling for so many years to develop a product, and before you go to the market, you have to establish your IP rights. What are the type of IP protections available? It can be trademarks, copyright, or patent. So trademarks, I have given you all the things. What is a trademark? What is a copyright? What is a patent? And the time limit is also provided here. IP rights provide the owners the right to exclude others from commercializing their creation, thereby giving them the monopoly over the market for a fixed period of time. So to create a monopoly, you need this IP rights. For protecting IP rights in academic setting. File a provisional patent application before publishing or presenting the result in the public. Scientists must take precautions in order to avoid the loss of patentability. Simple practices can be adopted to guarantee the novelty protection. Submission of abstracts and manuscript before patenting the content is possible. Also, it is advisable that a patent attorney draft and file your patent application because it is a legal document to ensure the maximum protection to your legal rights. Protecting your company or your name or logo via trademark helps you to deal with preventing unrelated business or organization of using your trade name. Do prior patent research during the initial stages of starting your research work. So this will help you in following ways: preventing waste of time on duplication of products, find the relevant information not found anywhere else, see what the competition is doing, and modify your invention according to the current requirement. So we should understand the most famous patent innovation known to human mankind is electric bulb. So we know it is Thomas Alva Edison in the year 1878. But however, an English inventor called Joseph Swan had received a patent for similar product in England at the same time. So there is a long patent battle for electric bulb. So researchers should realize the main reason for securing IP rights is to increase the chance that the invention is developed into a product, especially in pharmaceutical business. Decision on whether or not to develop a certain product heavily depend only on the existence of strong IP position. So we understand the path to eventual clinical application may not be a solo adventure. So it is a team. So we provide expert advice on IP protection, easier methods to adopt, and simple practices that allow free scientific exchange while providing proper background for potential business exploitation. Protect your invention in the right way. Please contact us for clinical trial, patent, trademark services, etc. So thank you for your patience listening my complete webinar. And in this corona pandemic situation, I would like to tell you, let us boost our immune system to protect us. So take good food rich in micronutrients, which will enable, or the immune boosters, which will enable us to boost the immune system. Thank you very much.
Thank you so much, ma'am, for your presentation. That was very thought-provoking and very well presented webinar we have ever seen. Thanks a lot. And we have come to a questionnaire session. Participant, if you like to ask any question to ma'am, please post the questions on the chat box that you have, a common chat box, so that ma'am will be honoring your questions. And before that, I would like to ask you a question, ma'am, like just in normal general question, like why haven't India came up with any of the corona vaccine yet? That are available to a common people.
We are developing vaccines. We are developing, the Serum Institute of India is developing the vaccine along with Oxford. Covaxin has come in picture from Bharat Biotech, and other vaccine has come. See, as I said, you cannot imagine within a day to come. It takes, you need to find out, you need to elucidate many safety aspects and the efficacy of vaccine before it is taken into the human beings. So time, maybe the time span, it takes time. As I said, go across the NIH site for clinical trials. You can see so many vaccines that has been in the picture. And Russia has said that they have developed into ultimately a product vaccine. So we have to come across that. So there is, yeah, India is doing very best of that. But only thing is, as I told you, it has to come across phase 1, phase 2, phase 3, and all these things. It takes time.
Thanks a lot, ma'am. I guess we have come to an end of the session, webinar session. Thanks a lot. I would like to thank you for very informative webinar, Dr. D. Sheela, Dr. C. Sheela Sasikumar. And I would like to thank Dr. D. Sudhakaran, GM of Golden Jubilee Biotech Park for Women Society, and Dr. Shamni for conducting this webinar and being supportive for us in behalf of Organogenic, and Dr. Ganesh Kumar Mani, who have been supporting for each and every session of us on behalf of Organogenic. Thanks a lot. And not the least, but you are the best participants. Thanks a lot for supporting us. And please do subscribe our YouTube channel. And this video presentation of Dr. Sheela Sasikumar ma'am will be there in a recorded version in YouTube, in our channel, YouTube channel. Thanks a lot for everyone for being there in the webinar session. Thanks a lot.